After the work of Hershey Chase, biologists were finally coming around to the idea that, based on the current evidence, DNA is what carried the genetic blueprint. But they still didn't understand how it worked. You take the work of Chargaff, and we started to understand the composition, but the actual structure still eluded us. Researchers such as Maurice Wilkins and Rosalind Franklin were studying the structures of different biological macromolecules. Rosalind Franklin was an X-ray crystallographer. X-ray crystallography is a scientific tool in which you take a crystal of your macromolecule. Here it's a crystal means that it was precipitated out as a salt. So you've treated whatever your macromolecule is, and it's now in a crystalline form. And they would take X-rays, a tight beam of X-rays, and they would hit the crystal. And then they would study the diffraction pattern off that crystal. And then they could calculate the overall shape of the structure that your macromolecule crystallized into. When Rosalind Franklin would take a crystallized form of DNA and would bombard it with that tight beam of X-rays, and then would expose the refraction pattern to, would study the refraction pattern on X-ray film, she noticed that this pattern kept coming up. A circular, my bad, not a very good circle. A circular outer pattern. Again, sorry, apologize for the not so great circle. And then this crisscross. This pattern kept coming up over and over. Okay. She couldn't figure out what the pattern meant, but this is what just kept appearing. Story has it that can't remember if it was either Watson or Crick, one of them, was visiting her lab and saw this pattern and asked to borrow some of her X-ray films. Based on the films and Chargaff's rule, for every A there has to be a T, for every G there has to be a C. Watson and Crick were already trying to build a model that met what specifications were known about the structure of DNA. Now, everybody knows double helix, Watson and Crick. And the question is always, well, if it was Rosalind Franklin's work, why wasn't she mentioned on the Nobel Prize and all of that? Well, it's one of those idiosyncrasies for the Nobel Prize. You have to be alive to be able to win it. Rosalind Franklin died a few years before Watson and Crick were awarded the Nobel Prize. Watson and Crick argued that, yes, it used some of her data, but it was their idea on how to interpret said data that allowed them to determine the overall structure. So based on the structure, knowing Chargaff's rule, knowing the individual nucleotide shapes, and knowing that there has to be an outer circle with a crisscross pattern in the middle of the structure, Watson and Crick were able to deduce the true shape of the DNA double helix. When you look at the DNA double helix, the red ribbon you see right here, we know is the sugar phosphate backbone. The crisscross pattern that was noticed in Rosalind Franklin's X-ray crystallography is the nitrogenous bases forming the rungs of the ladder. So you have that blue ribbon, which is the sugar phosphate backbone, which is the up and the riser, or not the riser, sorry, the up-down portion of the ladder. The rungs are the nitrogenous bases. Now, you don't just twist the ladder, no. What you do is you take the ladder and you wrap it around a pole, a central axis. That gives you this double helix, and when you look at it end on, the blue ribbons, the sugar phosphate backbone, forms a circle. Turns out that the nitrogenous rungs, the nitrogenous bases forming the rungs, forms a crisscross X pattern. Watson and Crick were able to build a ball and stick model to show, to illustrate what they were looking at, to illustrate their idea. Their idea had to take into account not only Rosalind Franklin's X-ray crystallography work, but also applied Chargaff's rule. And that actually took another great leap of logic. Other contemporaries of Watson and Crick were also working on this. Were trying, when they looked at Chargaff's rules, for every A there has to be a T, for every G there has to be a C, they took that literally. String of A's had to be followed by a string of T's. String of G's, there's going to be a string of C's somewhere in there. So what happens when you put this together, A to an A, G to a G? Well, that was either too wide or too narrow. Watson and Crick were able to deduce that based on size, based on shapes, that the only way you could meet not only Chargaff's rule, but also meet the constraints under Franklin's X-ray crystallography work was if A bound to T, G bound to C. So it wasn't A to A, T to T, it was A to T, G to C. By looking at this and examining this, their model met the parameters set by Franklin's work. Sugar phosphate backbone has to be able to form a perfect circle that meets a certain diameter, and that the nitrogenous bases had to connect together to form that X pattern. Watson and Crick were able to deduce based on shapes and positioning of the nitrogenous bases that adenine has 2 hydrogen bonds with thymine, and that guanine has 3 hydrogen bonds with cytosine. Now, there's been a big push as of late to increase the influence and the work of Franklin. Without Franklin work, there couldn't be Watson and Crick. And to minimize the Watson and Crick's work that everything they did, they stole from somebody else. You have to be careful with that. Watson and Crick, yes, they took some information from Franklin. Yes, they were a-holes who did not give her, enough credit during her lifetime, or even afterwards, after they won the Nobel Prize, they have argued against giving Franklin too much credit. But we have to make sure we also give credit its due. Watson and Crick were able to take the logic leaps to determine not only the structure, but the bonding patterns of the nucleotides.